Hi, everybody. Welcome to the last Optical Sciences Colloquium for this year. And when I say that, I actually mean the academic year. So you should all come back in fall. Yeah, it's not the year. So today we are actually very happy to have Roarke Horstmeyer here. Roarke is an assistant professor of biomedical imaging at Duke University. And he's also scientific director at Ramona Optics. So we will also talk a little bit about this, I guess. Roarke got his PhD at Caltech. And after that, he was an Einstein International Postdoctoral Fellow in Berlin. And after that, he was a visiting professor at the University of Erlangen-Nuremberg, also in Germany. So I asked him before if he can talk a little bit German. He said, well, he can buy some stuff at shops. But that's probably also, but we need to practice then a little bit. So so what Roarke is doing, and I think he will tell you a little bit more about this during his talk, of course. So he develops microscopes, cameras, and computer algorithms for a very wide range of applications. For example, he is doing like large area, high resolution 3D video of like 3D moving organisms. And he's also detecting blood flow and brain activity deep within the tissue. This is very interesting. Roarke, we are very happy to have you here today, and the stage is yours now.
All right, great. Thanks. Thanks for coming, everyone. Happy to be here. It's such a cool college program to visit and see. Optics is kind of the core of what I do, and so it's awesome to be here in kind of the mecca, I guess, of it. And so this talk is about some of the technology we've been developing over the past 5 years, 6 years now, really since I was in my postdoc time. And during that time, I was in a lab that was working a lot with model organisms. So those there's a lot of different model organisms, but smaller ones that are useful for microscopic study. Include the fruit fly, the zebrafish, or the Danio and C. elegans, which you may have heard of. And so a goal in studying these organisms is to uncover, their behavior, their genetic profiles, their morphology, how they interact with the world, how they interact with each other, right? And so you need to study them living, or it's beneficial to study them living. To do so, they're going to be typically moving around. And so you want to have a high-speed imaging system, and you want to see them at high resolution, ideally, to observe, for example, their fluorescently labeled cells or other small movements of their body. And to achieve these two goals, it's actually quite challenging in an imaging system, as I'll get into. And so there are existing systems to image at high speed in a microscope and with high resolution. And usually those systems involve tracking and following an organism around as it's freely moving to study its behavior and potentially measure its neural activity. Our goal in this initial effort to better capture and understand these organisms during their free movement was to try to image this very large area, to see all of this petri dish, for example, but at cellular resolution. And so that's tough to do, but we came up with a couple approaches. And then those approaches also were amenable to 3D capture. And so this laundry list of specifications is the technical sort of foundation for the technology we were initially trying to develop and have continued to develop. And so the question is, what kind of optical system can achieve all of this? And there's a number of different optical solutions. Naturally, you're probably thinking of some. But to put this in context of what exists, there are microscopes, at the very, very high resolution edge of things, pushing, everyone argues about resolutions in these super resolution microscopes. But anyways, tens to hundreds of nanometers that can potentially image in 3D. But their field of view is very small, right? They can only see a few cells, for example. As you move up this line, there's this middle regime, which people call the mesoscopic or mesoscale regime, which is micrometer, tens, hundreds of micrometers of resolution over somewhat sizable fields of view, right? So you can think of like a macro camera, macro photography type system, pushing the resolutions of that. And then all the way at the upper edge of resolution field of view, you have macroscopic imaging, right? And so for each of those categories, you have different technologies to measure in 3D. Macro, there's a whole host of methods, right? From LiDAR to stereo imaging to frequency modulation, which I've seen already today in a few labs. And then at the micro regime, typically 3D is captured by modulating the illumination, either with a light sheet or confocal, or generally even multiphoton approaches. In the middle regime, there's really kind of this funny lack of 3D imaging technology. And so that's some of the stuff we've been working on, as I'll get into. And so we're kind of on this curve here, and then our goal is to kind of push off and get a little bit beyond the standard trade-off between resolution and field of view to see much larger areas at high resolution. And so the problem, the origin of this kind of dashed line, or one of the origins of it, is caused by lens design, as I think many of you can appreciate being optical engineers. It's hard to design a lens that simultaneously gives you high resolution and a large field of view. It's not necessarily dictated by the sensor, which only has a limited number of pixels. You can get sensors now with a couple hundred megapixels easily, and it's not impossible to imagine that scaling to larger pixel counts. But the lens in the microscope really is a big bottleneck. So this is a table of this standard Olympus microscope objectives. They have different kind of suites of microscope objectives. This is just one of them. But they're all in the same regime where you have a resolution that goes up with magnification, but as the resolution goes up, the field of view drops proportionally, right? Because you're quote unquote zooming in more. And the product of those two, or rather the division of the field of view by the resolution, gives you the total number of pixels the lens can support or resolve. And that tends to be a constant in the order of tens to 50-ish megapixels, right? And so how do we work around that? So this is a picture of myself. Oh, there's interesting like Morier aliasing on this projector. And my co-founder, Mark, who doesn't wear shirts with those stripes. It's just an artifact, as you probably can appreciate. That that's next to a lens that has a throughput of several gigapixels. It's for lithography. And so there are lenses that exist that can resolve this number of spots. They're just really large and expensive. I will show a couple lenses later in the talk that can support a large number of resolved pixels that we've been working with. This is actually a lens from an old X-ray system that is mapping the X-rays hitting a fluorescent screen onto a large format film. And so there it has a very high numerical aperture to capture lots of the fluorescent light and can resolve over a large area. But these lenses are sort of few and far between. And you still need a sensor with lots of pixels, right? You're still left with that fundamental detection problem if you're focused on this single lens design. So our solution to this problem, which isn't a new solution, it's an adopted solution from others who have worked in this space, is to make an array of imaging systems. So the step one in our solution was to take a microscope or sort of a macroscope and shrink it down into a little footprint, which I'll show we can readily do. It's not too hard to imagine doing that, given the power of the imaging systems you have, everyone in here, in your pocket, in your smartphone. And then we put those in an array. In the array here as 24, we create arrays up to 96 of these, what we call micro cameras. And then in this design, each camera is imaging a unique spot on the focal plane. And we can capture, for example, this area with camera 1 in the upper left, this area with camera 2, and we can digitally stitch all those together if we want to form a large area composite, which allows us to see a much larger area at a high resolution, otherwise not possible with a single microscope objective camera design or microscope design. Okay, so like I said, this is definitely not a new idea. There's, arguably two ways to split up this camera array space, I would say. One is camera arrays that just exist and directly image outwards. And so those date back, several decades to camera arrays for light field imaging, which was a big effort at Stanford, for example. You can have camera arrays in astronomy. I'm not such an expert at that. I just know that exists. And I know amateur astronomers are purchasing these macro photograph systems and putting them in arrays to do surveillance, pervasive surveillance of large areas of the sky, for example, to find asteroids and things like that. There's camera arrays in our phones, right? So that's a natural setup that has this arrayed technique. And then there's multi-scale imaging. So Dr. Brady here is a pioneer in this field, and you can have many cameras in an array that image or capture light from an intermediate plane. And here it's imaging an intermediate plane. And these multi-scale imaging systems, have also been extended into the microscopic regime, particularly by this work here. It's called the Rush microscope, which used an array of many cameras and some big optics, and they were able to get really amazing large area imaging to see neural activity across an entire mouse brain at very high resolution. And so the work I'll present here is more, it's kind of spanning both these spaces a bit, but more the initial stuff I'll talk about is more in the direct imaging side, so the right side. So surprisingly, there hasn't been that much effort to do microscopy directly with an array of microscopes. And if you drew a little diagram like I drew here, you'd quickly see why it's not maybe the best idea originally. And so if you have a microscope, usually you're magnifying an image, right? So you have a spot in the real world that's being mapped, projected to a larger image where I'm drawing at the top an image sensor. And so if you have a magnification setup, you'll always be left, and you put two microscopes together side by side, you'll always be left with a gap, right? An area you can't see. And so that's not great for the original goal I set out to achieve. And so what you really need to achieve is high resolution imaging in a regime where your magnification isn't greater than 1, right? So it's 1 or below. And so in this regime, your pixel size really starts to matter, right? Basically, the best resolution you'll be able to get if you have a magnification of 1 is simply you're mapping your pixels into the real space. And so it's going to be your pixel size of, let's say, 5 or 10 micrometers historically that would define your resolution if all of your optics were perfect. And that isn't great for microscopes, et cetera. And that's an ideal case. It's tough to get these image sensors directly next to each other and all of that. But over the last 10 years or so, pixel sizes have dramatically shrunk. And so the pixels we're using in our systems now have 0.7 micron pixels. Previously, a lot of this work are with 1 micron pixels. And so in that regime, you're now talking more like in the resolutions, if you spoke to a microscopist or biologist, they'd be like, oh, I can work with that. And so it's really these arrayed approaches for the high resolution microscope imaging world are enabled by these very small pixel sizes. And so when you then put these in an array, you can operate these multi-camera systems in a couple different regimes. One is this sweet spot that I've shown where the fields of view of the cameras just perfectly overlap. So that's this green line. That's this kind of continuous field of view regime. Another one is you can zoom out more and any point in the scene will go to 2 or more of the cameras, right? And so that's this multi-view regime, which is going to get us to this 3D imaging. And then there's this third regime, which is more just like a bunch of microscopes all imaging in parallel, where there's gaps. That's what I'll just call a tiled regime. And so I'll show some examples of each of these regimes. But you can map this space out, and it's a pretty simple space to analyze. But the resolution naturally, as you zoom in and go to the tiled regime, goes up. So you can get higher resolution as you zoom in at the sacrifice of having to scan around. And then when you zoom out and you start to get multiple views, your resolution drops, right? It's not so surprising. We wrote a paper kind of about these trade-offs this last year. Okay, so here is our second generation camera array that we made. The specs are kind of laid out here. Our first generation array, I'm not even showing, but it was 96 cameras all packed together, but it was really limited to 1 frame a second. And so that wasn't that useful. It was cool, but it wasn't useful. So then we redesigned everything and made this so we can go 10 up to 240 frames a second. So really getting those high-speed videos that are needed for these model organism studies. The field of view is about the size of an index card. It's 8 by 12 centimeters. And the resolution for this system was 9 micrometers full pitch resolution. So not so bad, 4.5 micrometers per pixel type resolution. And so naturally, it's a lot of data to ingest. We're running this in a regime where we simply have to throttle everything to get data into memory. In other words, the overall limit that we face is caused by just the number of bytes we can get into memory. And so we were able to architect everything to go up to 7 gigabytes per second in this first system, which kind of put us in this 10 frames per second regime with the full resolution of around 700 or 800 megapixels. Yeah. Yeah, great question. So we controlled all of these cameras with an FPGA, or that's how we control it, yeah. And then the FPGA routes it all directly via PCIe to computer memory. Yeah. And so we can go either directly to SSD or RAM. Yeah, good question. Any other questions? It's good.
Yeah. Did you ever consider using event cameras?
A little bit. I've played around with event cameras, but they're a little bit, well, let's just say that cell phone type cameras are very inexpensive. So if you buy a few hundred of them, it's not a big deal. So that was maybe the easiest starting point for us. Yeah, it does not translate over there well. So it says the field of view is similar, 120 by 80 millimeters. So that's the overall field of view. The resolution is 9 microns full pitch. It kind of repeats what's over here, actually. The modalities are bright field and fluorescence, and there's live stitching. So we've kind of shown, we've created software like that can stitch the images together in a live manner. Okay, so this isn't just an example, really short video. It's hard to show these very large images, right? Naturally on a megapixel projector, there's like a gigapixel of resolved spots. But essentially, we can see anywhere in this field of view. This is a well plate. So a big application of this is to look at organisms or other specimens in what are called well plates. These are these plates that biologists routinely use and put different specimens in to study them. And we can see anywhere in this space we can capture at this type of resolution. And then we've been developing tracking software to follow these organisms around to report key statistics. And so some of the experiments, this is again, one well, but we are capturing 96 of these all simultaneously with this camera system. We can track this zebrafish larvae, and you can see these little black dots barely moving around, maybe. Hopefully, those are paramecia. That's what it eats. So we're observing its hunting behavior and then testing drugs, how those impact the hunting behavior, including Ozempic, actually. I'm not in charge of that study, but the collaborator is interested in how Ozempic impacts zebrafish appetite. Yes.
What makes it necessary to have the wide field of view that you couldn't do by rastering? Is it signals that are going so fast that it's faster than the rastering could do?
Absolutely. So in this example where they're all in different wells, the existing approach is just to raster one at a time, right? And so if you want to do a hunting assay, which no one really ever does, it's kind of a new assay, you want to watch this behavior for like 10 minutes. So if you're going to raster, you're going to watch 10 minutes at each of 96 wells. That's 960 minutes. And so these fish come in, they're like 3 days old, it says, 3 days post-fertilization. By the time you finish that raster, the first one was 3 days, and then the last one was 4 days post-fertilization because your whole experiment took a day, right? So your data is hard to interpret. And so essentially, the rastering approach in a lot of these high-throughput studies hinders video-based assays, hinders the ability to record and watch live dynamics, right? If that makes sense.
Well, individually, just over and over.
Yeah. That's what I guess you would, a standard approach would do with just a single microscope, if that makes sense. Yeah. And that just isn't, you have to go from well to well, and if you have to take a 10-minute video at each well, it takes forever. Okay, and so for these zebrafish results, we've developed analytics. I won't really dwell on this, to look at how they're moving over time. We then kind of distill all of this into these categories of behavior. And then at the end of the day, we simply present biologists typically with these type of results, which they really love. They don't love seeing gigapixel videos or 96 videos, right? It's too much. But if we simply can distill all of that information rapidly and accurately, which is, not easy, into descriptions of what's happening biologically, then it seems to be quite useful. So these are plots for different wells, the types of behaviors that were being exhibited that we're just automatically categorizing using machine learning methods. Right? And so you can see here, the fish were predominantly stationary, but for example, like if a certain zebrafish had expired or was really excited by a drug, then this blue part would disappear and it would become much more colored, for example. Right? And so anyways, this is a long way to say that we're ingesting for this plot probably like 10 terabytes of data. But at the end of the day, all that matters is something like, 500 bytes of information. But I guess that's just something we've learned is useful. So we can image in fluorescence in this modality where everything's continuous. It's not great fluorescence. It's okay. So it's kind of hard to see, but each of these is a zebrafish larvae in these 96 wells. And these are neutrophils that are labeled fluorescently that are moving throughout the body. This isn't a real-time video. This is like one image every 10 seconds. They are moving actually quite quickly, but just slowly moving for a wound healing assay where those cells are moving to go help with a wound. Okay, this is just another cool video that we took. Oh, it's kind of dark. Of again those zebrafish larvae here in a petri dish. And they all kind of shake around and get startled, which is kind of interesting. So this is just showing the continuous field of view type resolution. Okay, so moving on to more interesting configurations. Here, a second one is this multi-view scenario. And so when you configure this arrayed microscope to capture multiple views, there's a number of choices you can make. Do you want to have 50% overlap, meaning 2 cameras see each point in the scene, or 67% overlap, meaning that 3, and this is just in 1D, so naturally it's more in 2D. It's a design choice. And so when the goal is 3D imaging, we've actually played around a lot with this, and we settled on a configuration that loosely looks something like this. And you can see immediately that in our design, start changing the focus of these lenses, it's hard to get everything really tiled together perfectly, right? And so naturally, when we do these 3D recordings, we always have to capture a pre-calibration image. So we image something kind of patterned and fixed that we know. From that, we can figure out what camera is seeing which feature. It's pretty important. You'll also notice that when we do this multi-view imaging, the number of viewpoints is not fixed across the field of view. It changes. That's just because we have natural edge effects and imperfect overlap, right? But we've kind of settled on this type of design. It tends to be pretty good, where you have between 4 and 9 views for the majority of the field of view. Yeah. Yeah.
Did you find it was important to do it in the same plane you expected to image at, or was there an ideal?
Yeah, we need to do it in the same plane we're imaging at. Yeah. So generally, what we do is we have some area or arena or a place where we put the well plate, and we just put a target there that's, you know, at the same height as the wherever the base of the focal plane is going to be. Yeah. And so to get 3D measurements, it's a coupled problem with stitching. So we capture all this data, right? 54 pictures, let's say, and we want to stitch them together, for example, to create a composite 2D image. If things are at different heights, then the stitching won't work perfectly, right? Because, you know, if something's closer, it's going to shift, you know, when you open one eye versus the other more than if it's further away. And so how do you stitch that finger together without knowing its distance, right? And so to get really pixel perfect accurate stitching, you need to know the height at each pixel effectively for a non-flat scene. And so you really want to solve for the 3D height and stitching at the same time, right? When you need to do both. If you don't need to do both, then just solve for the one you need to do. But we need to do both. And so this is not a new area either in software and computer vision. This problem is called the photogrammetry problem, or the solution is called photogrammetry. I don't really know actually which way to say it. But photogrammetry is really common in imaging in macroscopic settings. There you see images like shown on the left, where your imaging systems are generally non-telecentric, right? So you get perspective, parallax distortion rather, at different locations on the imaging plane. This is different than in most microscopes, where most microscopes are really optimized to be very telecentric. Because when you're observing, let's say, a tissue specimen or something, you don't want it to appear different in the upper left-hand corner versus the middle, for example, during some review in the clinic or something like this. So the challenge of performing 3D imaging, or the way we solved it, really isn't amenable to microscopes, but it is to our camera arrays, which are non-telecentric, right? And so it kind of is an interesting space where perhaps if you're interested in 3D imaging and you want to do microscopy, I think it's beneficial to think about these types of non-telecentric microscopes. They don't really exist very much, but maybe they should. Okay, and so the way this works is we take a picture with one camera, and there's a vanishing point that I'm just keeping as stationary, right? And so if we shift the camera over to the right, you'll then take a second picture, right, where everything's shifted. And then let's say we look at, sorry, we're not really shifting the camera, we're just imaging from different micro cameras. If we image from the micro camera on the left, we'll see this picture, right? So these are 3 pictures we capture of a particular scene, and our goal is to stitch them together and estimate the 3D height. Visually, you can immediately see, because our brain is programmed to do this, that this blue tower is taller than the green and red one, right? Because it's stretching more, right, away from the vanishing point. So we're really good at doing that. And computers, you have to have them, teach them to get good at that. And so the process of teaching them to get good at that is something called computational orthorectification. And that involves shifting pixels, so picking each pixel and shifting it towards the center by a certain distance defined by this yellow arrow that is proportional to the height of the object, right? And so that's really all we're trying to do when we perform this procedure. We are shifting pixels in each image inward proportional to the height that we think it's at. Once we've shifted them inward, then they become telecentric and we can stitch them together, right? And so that is just simply happening together. And we use the metric of performance, of success, is simply our stitching accuracy, right? Are we getting better and better stitched images as we shift the pixels around? Once we're done shifting them all around, we have our height map, right?
Each object from the side walls.
Yeah, so the lighting effects, we're assuming a diffuse scene in this case, and the illumination's diffusely coming from above. We have an array of LEDs that we illuminate with. Yeah.
What about the occluded area?
Yeah, cleared areas are tough. Can't help you. We're not getting like a full 3D volume in this case. We're just getting a surface height. So I didn't invent this term either, but I like it. I call it 2.5D imaging, really, is this regime, right? It's like RGB depth type 3D. Yeah.
The objects as prior, are those jointly decided or?
No, we don't.
Oh, okay.
Our optimization algorithm is simply trying these different shifts out. It's learning those shifts to then get these to stitch better and better. Yeah, we've tried a bunch of different ones. Yeah, generally, yeah. I actually don't know the exact one we used in these results, but I could pull it up later. Yeah.
From Zoom, that if you could repeat the questions that people ask.
Absolutely. I'll do that going forward.
Yeah, next time.
Sorry. Okay, and so we went ahead and validated this with gauge blocks, and just to get a sense of the accuracy we're able to get with the height estimation. Arguably, so visually, right, you can see, right, we have a gauge block that's 50 microns taller than this one. This one's 20 microns taller. This one's 100 microns taller. You can see 100 microns visually, right, changing, but probably not 20 or 50. That's my gut feeling. We get lots of questions about this. If you look at the inner pixels and start to play games, you can claim accuracies more in the 50 micron, 20 micron. But in this architecture, I'd say around the order of 100 micron height accuracy, which isn't so bad, but can definitely be improved on. And so we've been developing more compact arrays, as I'll show, that can improve this height accuracy. But this is an example. So naturally, this is very amenable to video, right? We just hit play and we're recording all of these frames and we can capture these videos. Here are some ants moving around and you can see we can zoom in and then you'll see we show the height map. And so these are the heights and you can see then the error, which is why I love this rendering, that we're not getting them perfectly, right? So, but it's still pretty good. So we can begin to measure how their limbs are moving and as they're moving and interacting. And so, yeah, we were working with a few labs that study ants to examine their fore, mid, and hind leg movements. And we've also applied this to a number of other organisms. These are zebrafish larvae. You can kind of see we've numbered them all because we used tracking to follow them around to create these zoomed in versions. And so you can see these zoomed in versions compared to the resolution I was showing when we were doing the feeding assay. This is much lower, right? And that's caused by two things. One, we're zoomed out a little bit, which actually doesn't change the resolution too much. Maybe it's like a factor of 2 or something. But we're running this at 230 frames per second now. And so we're also doing pixel binning. We have to do that just because of the data issues, right? But here you can see these are centric views of each of the zebrafish, and then these are their heights. And the little specks are food, not the moving food, but stationary food, because the resolution for this wasn't high enough to see the paramecia, and so we did there's a word for it, I can't remember, but like the sprinkled food you'd put in a fish tank, but just a little fancier for this case. Okay, so we can then zoom in. This is the third configuration I was mentioning, where we get really high resolution imaging. We've been trying this with a number of different specimens and sample types. I'll show some examples from imaging well plates where we're imaging organoids with this, and then I'll show just a quick thing of slide scanning. But in this space, we made a system with 96 compact cameras. In this design, sorry, you can't really see. Let me just. These well plates are very standardized. They're always 8 by 12 centimeters, which is why we designed our field of view to be 8 by 12 centimeters. But also in a lot of them, the 96 well plates always have 9 millimeter spacing between the wells. It's just a standard. There's robotic systems that work with these. They inject things, they suck things out, right? It all just has to stay at that. So because of that, we designed all of our lenses to be 9 millimeters apart. And so each one could be centered on one of these wells. The lenses we designed, because this is an optics group, we've designed a few different lenses. The ones shown here are 0.5x finite conjugate lenses, which are actually pretty nice. I think they have like 9 elements in them. And yeah, they work really well. The frame rate, again, in this design is between 10 and 120 frames per second. And we were able to increase the data rates in this updated work to 10 gigabytes per second. A little bit better. And so this is just an example of some of the stuff we've been imaging. This is actually a 384 well plate. We could capture that by scanning in a 2x2 grid. But we've been imaging organoids. I don't know if others here work with organoids. They're really great specimens to image in the microscopic space. That you can grow them to all different sizes, and you can have them be fluorescent, and really interesting phenomena occur. So here, this is a brain organoid. These are little, 1 millimeter or smaller effective brains that they don't have vasculature or anything like that, but they have neurons in them, and you can have those neurons have genetic encoding to emit fluorescence when they fire. So that's what you're seeing there, firing neurons. This is a cardiac organoid. You can see it's moving a little here. That's its beating, right? So it mimics the kind of contraction events that our own heart goes through. And then here, this is a time-lapse image of tumor organoids. So you can begin to study the impact of drugs, treating them, seeing how white blood cells go after tumors and things like that. Okay, and so in this platform, we also do 3D imaging. I know the title of my talk was 3D imaging, so I had to explain. We also are doing it, but we're doing it very simply. We're just doing a focal stack for this system where we're zoomed in. We haven't done anything fancy yet. You can imagine there's lots of fun things to do that we're thinking about, but we've just been doing focal stacks. And so, yeah, you can do just the standard wide field focal stack capture in this setup as well. So we kind of capture data like this. These are human-derived organoids, and then we can begin to process it. And so one thing we've done recently, which I think was pretty interesting, is we image brain organoids grown from patients, actually. This is in collaboration with the lab at UNC. So you can take cells from patients and grow these organoids, which is why they're of such interest. And then these brain organoids were treated with a drug, and we were studying that. And so you can see here, this is the data that we capture. You can see, yeah, there's nutrients in the well, and there's other stuff that make it not so easy to measure or see these organoids. And so what we did is we applied just segmentation software to find and define the boundary of each organoid after doing a 3D focal stack and then doing a projection to get a kind of all-in-focus projection of that. And we just use segment anything. So I'll also say I'm a big fan of segment anything. I don't know if you guys have used that yet. It's Meta's segmentation tool that's kind of like a foundational model for segmentation. it works great. We just annotated like 10 of these and it was off to the races. And then we're plotting here the area within this segmented boundary of these organoids as a function of 12 days. Day 1 is actually they're 2 weeks old, so that's when they grow to that first size, and then they grow beyond this. They actually can grow up to a year old. But this is just a test of a drug that's used to treat infants. Anyways, won't get into the biological details. It's an interesting drug used to treat brain disorders in infants. And from day 1 to day 12, when the drug is applied on day, where is it? Drug added on day 3, you see the brain organoids were all growing. Day 3, the drug's added, and then they all really respond to that treatment. And then each of these is a different dose of the drug. So small dose, a little bit more, etc. So yeah, we were happy to see this ability to record in parallel was actually useful in this brain organoid setting with that lab. Okay, we also designed, just because I want to show the optics, we designed other lenses. So this is an array of 48 imagers, also at 9mm spacing. And we worked with a company to design 2 types of finite conjugate objective lenses, actually. One is a 0.3 NA, and one's a 0.5 NA compact microscope objective. And what's interesting, it is finite conjugate, right? Both of them. So the 0.5 NA on the imaging side, or sorry, on the image plane, or sorry, the object side and the image plane side is kind of similar, right? You need like a 0.3 NA to image to tiny 1 micron pixels. But on the 0.5 NA, yeah, you have a larger magnification. I think the magnification was something like 4. Okay, so we use this to scan slides. This is still ongoing work. We put 3 slides down, and then this is just a video of the data we capture. And you scan them out. We're also scanning in 3D because these are thick cytology specimens, so they have a really curvy surface. And then we just do a projection. But this is just an example of the type of data that we're capturing. So yeah, this is a ongoing project, like I said. We have one of these at the Duke Medical Center. We're scanning a bunch of slides to help with some of their workflow. Okay, so for the last 15 minutes, I'm going to show more of the not direct imaging, but the multi-scale type imaging, or just playing around with camera arrays optically. So one of the designs we were interested in is something called a Fourier light field microscope. So this Fourier light field microscope is easier to explain in maybe one slide, but we made one of these with our array. And so to do this, we took a really big lens, the one I showed you. That really big lens is mapping light from a sample into, some sort of infinity space. I won't really call it an exact infinity space, but sort of an infinity space. And then these lenses are all focused at infinity now. So they're like imaging really far away to capture all of the light coming in. This will become clear in the next slide. But yeah, I just wanted to say this is done by a PhD student in my lab, Claire, who's great. So the way it works is essentially as follows in a Fourier light field microscope. There's a lot of kind of confusing description of disease. I think it's easiest just to understand by thinking of 3 points. So let's say you have 3 points in your scene. If you took a Fourier light field microscope and took a picture, each camera in your array would just see those 3 points, and they'd all look the same, right? If it's at the object plane of the imaging system. If it moves away from the object plane, in the middle camera, those 3 points won't move. But in this camera, they'll just shift, right? And so what you're left with is an imaging system where things shift due to how far away they are, right? Shift relatively across the cameras due to how far away they are. And so if you take some points, and this is experimental data, and you just move them away, and as you're moving them away, they turn from blue to red, then you capture data like this, right? Where each depth is encoded by the blue to red color, you just see this kind of expanding image effectively, kind of expanding laterally outward. And so you can use that information to just recover 3D depth, not surprisingly, right? If you look at one point in the scene, you can see how it's shifted, in relation to the other cameras and back out its 3D position. Okay, and so we've put together the system, and here's an example of a dinosaur. And I didn't know this, but at a talk a few months ago, I saw Dr. Brady had a similar dinosaur for some of his cool 3D imaging work. So I was really happy that somehow the 3D imaging community, at least us, we've converged on dinosaurs as the metric of performance, plastic dinosaurs. But yeah, the resolution's quite nice, right, compared to other systems where you capture light fields, where you generally have to take your focal plane of pixels and somehow split them up to sacrifice spatial resolution to get this light field type measurement. Here, we're also doing that, but we just have a lot of cameras. And so we're left with still a 13 megapixel image, right? Because we have 48 13 megapixel sensors in this design. And so you capture one image from the middle that looks like this, but then the image to the side looks, significantly different. The angular perspective shift with this lens it's 39 degrees in X and 30 degrees in Y, so it's a pretty large shift. And so you can just see visually, we get the decent perspective there. And then we do 3D processing. Sorry, this was from a talk I gave where Claire was talking after me. I should have just put up the 3D processing method, but I can maybe touch on that later if I have time. But this is the kind of 3D results that we get. And where this is really useful is in, or one area that we're getting requests for this type of measurement is in microsurgery or surgery where you need resolution that's slightly higher than what our naked eye can offer. And that's common in human surgeries, but also with model organisms. And so we've been working with a lab who perform surgeries on rats and also other organisms, but primarily rats. And they want to be able to drive a robot in 3D accurately to automate these injections and other manipulations. And so we've been imaging the skull of the rat. This is an example of a 3D image of that rat skull that we captured. Sorry for scale bar didn't transfer over. But it's quite large, actually. It's on the order of maybe 3 centimeters long is the scale that we're kind of observing here. And then we can capture, this is just a test. This isn't an actual surgery. Whoops, sorry, that was just the 2D image. But here's the tool in 3D kind of moving and touching the surface. And so you can see, you were speaking about occlusions, I think. You can see we can't solve that problem with this design yet either, or not naively. Sorry, let me play that again. Where's my mouse? You can see beneath the tool, we're not able to fill that in because we don't have enough perspective views to capture that. But what we can do is track the tool in 3D. So here are just 2 points on the tool, the green and red dot, and then this is their axial location, right? And so we've been doing experiments by moving that with a little stage and measuring our accuracies and tracking the 3D position and angle. And it's significantly higher than we thought. It's on the order of 5 microns in axial positioning, which we were surprised by. So we're really trying to figure out why is it so accurate. That's a separate story. Okay, and then the last manifestation, but probably the most interesting for 3D capture is trying to capture many, many more angles, right? So I just showed results where we captured 30 to 40 degrees of angular perspective. Ideally, we would somehow just image all around some specimen, right? If we really wanted a tomographic measurement to get full 3D. So we've thought a lot about this, and we just haven't implemented it yet. I don't know, maybe we will. But it was just hard, and we had these camera arrays laying around. And so clever postdoc of mine, I guess also, I worked with him when he was a PhD student too, Kevin Joe, came up with this idea of just putting a parabolic mirror beneath the array, and then creating effectively a 4F type configuration with the mirror as the second lens. And so here again, we're using each of the cameras, they're focused at infinity. And then the lens is acting as that second lens in the 4F system, so to speak, to then bring, map that light to a closer focal plane. And the focal plane will always be, within, passing through the parabola foci. Right? And so effectively what occurs is you get many, many perspectives from these cameras of this specimen that's placed at the parabola foci. And here's a kind of rendering that maybe shows a little bit more what's going on. And so this method, originally we pursued it just for fluorescence imaging. We're now starting to do bright field imaging, but you need to use some tricks to do that, because when we first tried just direct imaging, you got all these crazy caustics and weird kind of funny things caused by the mirror. And so when you have a fluorescent sample, it's just emitting light and then going to the cameras. And so a lot of those caustics are mitigated. And so then we need to illuminate the fluorescent specimen. And so the bowl has a little hole in the bottom, and we just kind of shoot the excitation light up in there. And then we have emission filters over the lenses to block that blue excitation light, and we just capture the green light. That's the kind of rough design. And so our reconstruction algorithm for this is we have a couple actually, but one of them for the results I'll show essentially models each voxel so this is a fully 3D measurement technique, so we don't have this problem of occlusions anymore. In this method, we model each voxel in our 3D reconstruction as a source, F, and emits green light, but it also is going to absorb light. If there's something absorptive, the green light will be attenuated. And so we need to have both those parameters in our model. We assign those, and then we have essentially a kind of ray-based description of that radiation passing through the volume, where the sources accumulate along a ray, and then absorption attenuates in the exponential, right, due to like Beer's law type absorption. And that will generate some intensity on our camera. And we compare that intensity on our modeled camera to the one we measured, and we minimize the mean squared error of what our measurements are versus what our prediction is with respect to these values, right? And then we cross our fingers. No, it works, trust me. But you'll see. But we actually figured out a kind of better way to do this, is to use a neural network. And some of the other results I showed, I didn't get time to really talk about it. For example, the 3D ant imaging. We're also using an untrained neural network in that approach, for some of those results. But this approach is sort of, we've been adopting it more and more for our different imaging applications and reconstruction applications, rather. But in general, what we found is instead of directly, so before I said we're minimizing the mean squared error loss or some loss function with respect to these values that we want, right? So instead of having those be the objection function variables that we're minimizing loss with respect to, we've found it much easier and more effective to put a deep neural network, a convolutional neural network into our pipeline and then minimize the weights of the loss with respect to the weights in that neural network, right? If that's an option, if somehow we can squeeze that in there, it tends to work much better for our imaging applications we've learned. And so for this approach, what we've done is we've essentially adopted a NERF type reconstruction method, or a neural radiance field type reconstruction method, where we have points in the scene, X, Y, Z, and time. Those pass through a neural network to give us an associated absorption and fluorescence emission value for that voxel at that position in time. Those we then create our arrays and do the exact same thing to form a simulated image, a pixel. And then our loss, our mean squared error loss, comparing our data to our prediction, is simply a function of the weights of the neural network now, instead of the actual, absorption and fluorescence emission values. And so doing that has this nice benefit of we're able to kind of get this network going, and then we can, for one frame, which takes a while computationally, and then after we've done that, we can fix this network effectively and then use it for all subsequent frames. So it gives us this speed up of processing time. Whereas before, each frame we had to process sort of individually. Here we can kind of use this bootstrapped approach. Okay, so this is what the data looks like for a fluorescent zebrafish larvae. Again, it's hard to show this data. Each of these is a 13 megapixel image from a different perspective. Here, I'm kind of zooming in on some. This is just one of a number of example fish. You can see that we have a number of distortions that we have to deal with. This is primarily due to the cuvette, or essentially it's an NMR glass tube. But the tube that's holding the fish introduces distortions we have to account for. But we can do that, and this is an example video of this zebrafish larvae, where red is showing its absorption, primarily in its eyes, right, but also elsewhere in its body. It has pigmentation on its skin that's absorbing light, and then green is the fluorescence being emitted. And so it's swimming around naturally, trying to get away from the bright blue light, I think, that we're shining on it. But it's swimming, and you can see it swam up out of the volume of view. It's in a long tube, so it can swim up and down. But we're beginning to try to move from this preliminary demonstration to actually showing some useful functional fluorescence measurements of brain activity, for example. Okay, and then we can analyze this data. I won't really dwell on that because I'm kind of running out of time. But we can track it and see what it's doing and things like that. We've done this with a number of organisms. So this is a fruit fly larvae with labeled what are called pericardial cells. That's the raw data. And then this is the reconstruction. You can see it's kind of squirming around. And what we were doing, what the lab was interested in doing that we were working with, is tracking how those pericardial cells move closer and further apart as it's squirming. And then the last one I'll show is, this is also a fruit fly larvae, and you can see a slow ripple here. That's this event or biological phenomenon called peristalsis. It's the same thing our throat does when we swallow. It's a very kind of fundamental biological process that worms, many hundreds of millions of years ago, have started to do, and now we do. And this lab was interested in studying those, and so we've been creating these plots of, the radius and also the rippling, which you can kind of observe here as a function of time, by measuring around that volume and watching it contract over time. Okay, so that's it. I will thank everyone who did this, particularly, like I said, Claire and Kevin were really instrumental in those last two projects. And then Ramona Optics, right, is commercializing this array technology, which you had mentioned. And so there's a team at Ramona which have been completely instrumental in getting all of this working. Couldn't have done it without them. All right, thanks everyone.
Thank you very much for this super interesting talk. I am prioritizing a question from Zoom because actually I have the same question. So structured light. So and probably other people have this question as well. So you can do structured light super resolution, for example. You can also exploit the same structured light signal to do 3D imaging. Have you considered using that?
Yes.
Hmm.
We just don't have time. I haven't had time. I'd love to.
Yeah.
It'd be great. It's a very natural fit. So
Okay.
I'd love to incorporate some structured light.
Okay.
Images you showed of those little test blocks,
Yeah.
with the stereo microscopy, the range resolution is going to be dependent on the object structure and texture, right? So really, you're getting range just from the edges there. So if you
Yes.
had it seems like you'd get much better if you had more texture, which you could give the structure.
Yeah. Yeah. Great point. Yeah. So that's why claiming some sort of resolution in that regime, I'm just a little hesitant to do because it just is very much object dependent. But if you resort to something that you're controlling, then yeah, absolutely.
Actually, you probably don't have to project a pattern or something. You can even do like some LED array stuff.
Yeah.
Right?
Yeah.
The slide where you showed the dinosaur.
Yeah.
That was really interesting. Oh, thank you for the talk.
Sure.
No.
Go back to the dinosaur. I like the dinosaur.
And so it's like, yeah, this is really interesting how you recover the 3D. But I noticed, since if I imagine a point on the dinosaur as a point source,
yeah,
you're getting specular effects, and yet so you should be able to do a synthetic aperture on top of this. As it looks like there's no specular effects when you move around the dinosaur.
Yeah, that's a good point. So, sorry, I didn't know it moved. Unless you guys really want to be hypnotized. So it's really like if you're familiar with light fields, that's essentially what we're capturing. So we can do like what's called digital refocusing through, and when you do that, you can then play games like pulling out specular versus non-specular reflections, things like that. So.
That's what I understand.
Yeah, I maybe have a slide, but let me just see. It's a little bit more nuanced of an approach than, I don't have a slide, but we can talk about it later. Yeah, it's not so simple.
Arrays of lenses, well-designed lenses for microscopy. Did you consider the possibility of using arrays of fiber bundles? Because you could construct lenses at the tip of one end of the fiber, and then the other end you can separate them from each other, so you can with multiple cameras on the other end. What are the advantages or disadvantages of doing one to start?
Yeah, so kind of using a fiber bundle array to map lots of pixels to our sensors somehow. Yeah. It just seems a little tricky to do that. So we've done a few fiber bundle array projects, and I think in certain regimes they're really useful. I haven't worked in one where you have to kind of take a large fiber bundle array and then split it into these smaller ones. Yeah, I don't know how that would work, but I'm sure it's possible. Yeah, it's a good idea. Yeah.
I had a question. So I've attended a few talks about like the zebrafish imaging thing before, and I remember that there was a particular discussion about like some like previous one microscope imaging systems that like ran into this problem of like with one microscope you have to like confine the zebrafish, so you get like, sad angsty zebrafish that like does all the things that are not helpful for science.
Yeah.
So then like I remember like the reason why we wanted to expand the region so that the zebrafish would be like a happy zebrafish that like actually went hunting and stuff like that.
Yeah.
So like but then like it kind of in a few of your imaging systems, like especially that like 3D one near the end, you mentioned that like you had some effects with your imaging system that actually did have an effect on the zebrafish behavior. And so like, I guess, how do you know that you have an index card sized imaging system at the beginning? Like, how do you know that it's big enough and that it's not affecting the zebrafish?
Yeah, I mean, it's an excellent question. Or I guess Oh, I'm supposed to repeat the question. No, maybe. The question was, how do we know the zebrafish behavior is not impacted by our field of view, if I have to summarize. And the answer is sometimes it's obvious and sometimes it's not. So we work with one group, for example, that doesn't work with zebrafish, but a very similar fish called a Danianella. And that fish actually vocalizes. It makes a small noise, has 4 sounds it makes. And if the arena is smaller than something like 30 by 30 centimeters, it won't make the noise. And if it's larger than that, it will. So like that's an obvious binary like kind of scenario. But naturally, there's all these middle grounds where you're testing drugs or something, trying these different things, and you're using some behavioral effect, like how fast is it swimming, and does that matter on the size of the field of view? And I think it's a totally an open question. If the drug is like a seizure-inducing drug, then I would argue no, right? It's just going to have a seizure no matter where it is. But if it's a subtle effect, then yeah, of course it will. And so yeah, those questions are really tricky. Yeah, it's a good, yeah.
Quick question for some of these like biological applications. I'd assume that synchronicity between your cameras is important. Like you're gonna grab frames from all 96 with some tolerance. How did you handle that? Like, do you have any tips or tricks?
Get a giant FPGA. That's what we did. Yeah, so these are synchronized up to 6 microseconds, which is just the FPGA's electronic tolerance. You can it's a whole, yeah, there's many things you could do. But yeah, there, it really depends on the synchronization accuracy you need. Yeah, so other approaches are you could have like master-slave configurations with your FPGAs and have the clock drive, groups of 4 or 6 or whatever. But we just took the kind of brute force approach of just one big FPGA. Yeah.
I think 1 or 2 last questions.
Thank you. Again, on the zebrafish, when you guys did the tomography,
yeah,
I guess it's CT, right? Did you apply filtered back projection when you did the reconstruction?
Yeah, I mean, essentially, this algorithm I outlined is a spatial domain approach, right? And so directly this. So I'll also say that we have 2 pi steradian coverage, right? We have like very full coverage. There's no missing cone really for that half of the hemisphere. And so in this approach, no, but then when we shift to this approach, and we wrote a paper about this in a separate context, the use of this kind of acts as an implicit filter, the use of a neural network, untrained neural network.
The reconstructed views matching the camera.
Yeah, it worked great. But this approach, or where is it in this step? The mapping from our kind of volume to our camera measurements is really important. We have to calibrate that. Yeah. So we have a calibration procedure for this as well. Yeah.
I have a question, and we should stop. So you talked a lot about occluders, and that you cannot get information when something is occluding. So there's all this light field work, where you look through occluders. Like
yeah,
how,
yeah. Huh. I never really.
Can you implement that, or?
Probably, yeah. That's a good point. But the occluders I was speaking about are like what we're imaging.
Yes.
So that's like a bush in front of the camera, and you want to image what's on the other side of the bush.
Yeah.
So in the microscope, if there's a bush in front of your microscope, you can just move it because that shouldn't be in your lab. But yeah, maybe there's like tissue or other things, and you're trying to see further beneath it.
Yes,
Yeah,
that was what I was thinking.
Yeah,
because
perhaps we haven't tried this.
Showed like I think that's so big in the field of view that you really cannot do something, but if you have like more like a bush kind of thing occluder, then
it's a good thought. Yeah, interesting.
Okay, so any other questions? No, then let's thank our speaker again.